Okay, well, hello everyone. Good evening and welcome to the American Cetacean Society San Francisco Bay Chapter's first speaker event for 2025. I'm Susan Hopp, board member responsible for our speaker program, and with me is fellow board member Wade Cobb. So first, for anyone new to the American Cetacean Society, we are a chapter within a national organization that is the oldest nonprofit dedicated to the protection of whales, dolphins, porpoises, and their habitat. And we do this through education, community engagement, and awarding grants towards cetacean research. We appreciate your donations in support of our mission. Donations fund our research grants and expenses for these talks. It's easy to donate on our website if you haven't at the time of registration. And speaking of your donations, we will be very excited to announce in February our grant awards for 2024 in the name of marine biologist and former ACS board member Izzy Shapaniak. It's truly your support in action, furthering research and conservation for the cetaceans and marine life that we all cherish. So thank you. So that brings us to tonight's presentation, which we are recording. Please put your questions in the Q&A and we will get to them after the presentation. Our talk tonight is Monterey Bay National Marine Sanctuary, Discoveries and Surprises by Chad King. And I cannot think of a better way to begin 2025 than exploring the wonders of our Monterey Bay National Marine Sanctuary with a scientist who has made his career exploring it at all levels. Chad King is a NOAA Research Ecologist with the Monterey Bay National Marine Sanctuary since 2002, and for the last many years has been in charge of the sanctuary's deep sea research. He's also responsible for the collection, analyses, and dissemination of spatial data for MBNMS and the Sanctuary Integrated Monitoring Network, known as SIMON. This data helps integrate past and present monitoring programs within the sanctuary and are the foundation of decision-making tools such as interactive maps that are made available to the general public. He is also a NOAA Divemaster and an active participant in subtidal research, including kelp forest and invasive species monitoring and underwater photography and videography. And additionally, he produces short outreach films and has produced significant content for the Sanctuary Exploration Center. Chad was instrumental in developing SeaPhoto, an iOS app that features imagery and life history content of the Monterey Bay National Marine Sanctuary. Also, a chapter in the new celebrated book, Wild Monterey Bay, by Jody Friediani and Caitlin Taylor, is dedicated to Chad. A native of the Bay Area, Chad earned a Bachelor of Science in Marine Biology at the University of California, Santa Cruz, and a Master of Science in Marine Science from Moss Landing Marine Labs. Chad, thank you so much for being here, and I hand it over to you.
Thank you, Susan, so much. Thank you for everyone for attending tonight. I'm kind of excited to talk about a lot of the stuff that we've been working on over the last, 4 or 5 years. So as Susan said, I'm Chad King. I'm a research ecologist here with Monterey Bay National Marine Sanctuary. Been here since 2002. Prior to that was with Fish and Game, and yeah, let's get right into it. I'm going to talk just about a couple of main projects that I work on. I consider myself, just as I start off here, just anecdotally, I love marine science. I love scuba diving. I love the deep sea. I love the near shore. I love everything marine science. But my expertise is very spread. I would consider myself jack of all trades, master of none. But I am a little bit better versed in some subjects more than others. So yeah, I'm involved in a lot of different things here, from diving. I'm a NOAA dive instructor, to microplastics, to field work, to iOS development, to website development, to photography, to all sorts of things. But that's what I love about my job. I get to do a lot of different things all of the time. And, it definitely beats a 9 to 5 in a factory punching the same thing over and over again. So that's what I take my pride in, serving the American public as a government employee. So I'll go ahead and get into it and talk a little bit about a couple of discoveries we've made over the last few years and some of those surprises and what they mean ecologically and to all of us. Just a little bit about myself. I kind of gave you a little bit of a preview, and Susan talked about it. I got my bachelor's at UC Santa Cruz, my master's at Moss Landing, worked at Fish and Game for a few years before I started here with the sanctuary in 2002. My expertise, and those are in air quotes, are kelp forest ecology and invertebrates. An avid scuba diver with the sanctuary, with over a couple or 1,200 dives or so. Uh, the last 6, 7, 8 years, I lead all of our offshore and deep sea research expeditions, and then I'm a GIS guy too. So I do a lot of our spatial data and database stuff. And on the side, I have a wonderful family, married with 2 kids. I have a daughter at she's a sophomore at Berkeley, and I have a son who's a freshman in high school. It's great to share a lot of this stuff with them, especially when I can take them to the aquarium and stuff. And on the side, I do feeding shows at Monterey Bay Aquarium every 2 weeks, so I get to jump in and talk to the public from inside the tank. And so my kids have grown up getting spoiled with their dad, being inside that tank. So, Office of National Marine Sanctuaries is large, and we have over 17 national marine sanctuaries, several of them designated in the last few years. In our neck of the woods, we had Chumash Heritage National Marine Sanctuary, designated in just December. So we now have 6 along the west coast, but we have others as well. And so it's a whole network of marine sanctuaries across the United States. Specifically at Monterey Bay National Marine Sanctuary, we are contiguous with 2 sanctuaries to the north, being Greater Farallones and Cordell Bank. And we have the new one to the south, who I just mentioned, Chumash Heritage National Marine Sanctuary. It's not quite contiguous. There's a little gap near Morro Bay, and then Channel Islands to the south. But as far as our sanctuary is concerned, it was established or designated in 1992. We have almost 6,000 square miles, and it's a wide variety of habitats, 276 miles of shoreline. We get to over 2 miles deep. And then, as I specifically point out here in the lower part of the slide, Davidson Seamount was added to the sanctuary in 2008 for reasons I'll get into in a little bit. Just as a primer, I even talked to the kids about this stuff. And I know most of you know this stuff, but I just kind of wanted to review and kind of get that 10,000-foot view. But 71% of the Earth is covered by water. We know this. We are a water planet, and the oceans are very important to us, our health, our survival. If it wasn't for oceans, we wouldn't be around today. However, if you think about it in terms of volume and living space on the Earth, because animals are living within the entire water column of the ocean, it's really 99% of the living space on this planet is in the oceans. Yeah, birds can fly, insects can fly, but they don't permanently stay in the air. So when you really consider about the ocean, these are animals, especially the planktonic animals, the midwater animals. They're there for their entire lives. So that is living space, and so 99% of it is there. And then, of course, 95% of those oceans are unexplored. And I'm going to get to this a little bit later, but Davidson Seamount is one of the best explored seamounts in the world, and yet we made major discoveries there. And I think it just really reinforces the need for exploration and the negation of assumptions that we know everything, because we certainly don't. So I'm going to go through a series of slides of relative depth here, if you will. At the bottom of the slide is the deepest part of the ocean in the Mariana Trench, which is just over 11,000 meters. And of course, the top of the slide is the surface of the ocean. Well, recreational scuba, which is what I do most of the time here, a lot of the time here at the sanctuary, we really can't safely go beyond 130 feet or 40 meters. And we're regularly much, much, much shallower than that. We never get over 100 feet, except once or twice every 10 years, honestly. but it's basically a red line at the very top of your slide. That's what we're exploring with scuba. It's nothing. It's a sliver of how deep the oceans can get. The Eiffel Tower is just almost 1,200 feet, 350 meters, and you can see the red line there. That's the relative depth to the Mariana Trench. Then we have a nuclear submarine, and I watched Hunt for Red October and those kind of movies when I was a teenager, and you think of it as these submarines going very deep. Actually, they don't. They don't go very deep relative to how deep the ocean is. Sperm whales, one of the deepest diving marine mammals, goes to about 1,000 meters. It's still not even 10% of what the Mariana Trench is. We have the Grand Canyon. Everyone knows about that. It's still only 1,800 meters. Really not that deep compared to the deepest part of the oceans. And then we have the average depth of the ocean, which ironically, or kind of almost counterintuitively, is not even halfway down as deep as the Mariana Trench at 3,690 meters, just a little over 12,000 feet. And then the deepest part of our sanctuary, Monterey Bay Sanctuary, is just slightly deeper than the average depth of all the world's oceans. And then even if you took Mount Everest and inverted it and put it down over the Mariana Trench, you still wouldn't hit bottom. So the oceans can get quite deep. And overall, when you look at our sanctuary here, it's that black line with the box over Davidson. 71% of our sanctuary is considered deep sea, and that's anything greater than 200 meters. The shelf is basically anything from 30 meters, kind of scuba diving depth, to 200 meters, and then the red or pink area is like scuba diving depth. It's the near shore. So only 4% of our sanctuary is really explorable by scuba. And really, most of that is up near the Golden Gate, which I'll be honest, no one ever scuba dives up there. So when you kind of eliminate the mouth of the Golden Gate, it's a very small sliver where we actually scuba dive compared to the volume and area of our sanctuary. So what I want to get to tonight really is work at the Davidson Seamount, two different projects. And the first one I'm going to talk about is a deep sea project. But the Davidson Seamount itself is one of the largest seamounts on the west coast of the US. It's 26 miles long, which is the distance between Monterey and Santa Cruz, if you're familiar with Monterey Bay, and it's 8 miles wide. However, it's about 8,000, just short of 8,000 feet tall from base to summit. So think of it as like if you're familiar with the Bay Area and the Sierras going over the Donner Summit. That's how tall Davidson is, Donner Summit height. The thing is that the summit is still 1,250 meters below the ocean surface. So the entire seamount is in perpetual darkness. There is no sunlight that reaches there. So there are no corals. There are corals, but there's not corals that use sunlight, right? It is an extinct volcano, but no worries. It last erupted almost 10 million years ago, so we're not worried about that. And now, relatively, it is one of the best studied seamounts in the world, but still relatively unexplored to the rest of the planet. So the Davidson Seamount itself has been a wonderful source of discovery, and I call it kind of the Dr. Seuss land. I mean, look at this slide of colors, your pinks and yellows and greens and all sorts of colors down there. It really does look like from a Dr. Seuss book. Anecdotally, back in 2000, we had a fisherman dredge up some of these deep sea corals, and even part of an old airship that I won't get into today, but it was from the 1930s. And they realized, oh my gosh, there's some really cool stuff down there. So in 2002, Mbari mounted an expedition with us, Monterey Bay National Marine Sanctuary, to go explore with their remotely operated vehicle, basically a robot that could go in the deep sea, and started observing all of these deep sea corals and sponges. I participated in the 2006 expedition, and that was in concert with the BBC, in which they got footage from that expedition and published it, and it aired in the first Planet Earth series, Planet Earth 1, their ocean series. And so that was really cool to hear David Attenborough talk about all the work that we had done and all the amazing discoveries we had made back in 2006. This was all a genesis for inclusion of the Davidson Seamount into our sanctuary in 2008. Well, back in 2018, I was tapped to be chief scientist for an exploratory cruise to Davidson Seamount. And really, the spirit of it with the Ocean Exploration Trust on their exploration vessel Nautilus was to explore areas that had never been seen before. Well, we had discovered a lot of these corals and sponges on the summit and flanks of Davidson Seamount. Now, even though it was just a sliver, a fraction of a percentage of the entire seamount that we had seen, we considered that explored. I know it sounds funny, but it's true. It's like, well, we've seen the summit, we've seen some of the slopes. Where else can we look? Well, within the confines of our sanctuary boundaries was kind of the base, the foothills of the seamount. And these areas I've called out here in red and yellow, you do see some structure there. Now, these structures are kind of resolved by remote sensing equipment called multi-beam. Basically, it's shooting sound waves to the seafloor, and then those beams come back up to the receiver in microsecond differences, right? And so those microseconds determine or can be calculated for different depths. You can use those different depths to kind of create a virtual image of the seafloor with shadows and everything. So you can kind of see the shape of the seafloor, which is really cool. It's kind of like one of those pin cushions, those little blocks where you put your hand under and push up, and you can see the shape of your hand. It's not your hand, but it's the shape of your hand. That's kind of what this is doing in a remote sense. So the idea was I selected these areas because they looked kind of rugose, meaning they had some cliffs and some slopes and everything like that. And the idea was we want to see what kind of corals and sponges are down here. And when I chose these areas, other geologists and biologists told me, "Oh, Chad, you're not going to find anything down there. It's just gonna it's that's where all the sedimentation happens. That's where yeah, you're going to find a few corals and sponges there, but they're going to be small. They're all going to be, covered in sediment. It's going to be nothing like the slopes of the seamount, where these deep sea currents rush up the slopes of the seamount, feeding these massive corals, some of them getting to 10 feet wide, corals and sponges as big as your living room sofa, which is true. And I've seen them firsthand, as you'll find out. So that's what I expected. But part of our mission as a sanctuary and what's in our management plan, which was guided by the public and the government, is to explore and characterize the resources of our sanctuary. So it was important to look at areas that had never been seen before, even if we thought they were going to be boring, because you never know what you're going to find. So we partnered up with Ocean Exploration Trust, which was headed or is still headed by Dr. Bob Ballard. And of course, if you don't know who he is the man who discovered the Titanic in the 80s with the Alvin, the submersible Alvin. And so he spun a lot of that notoriety and fame and expertise into Ocean Exploration Trust. And we partnered with his trust here to take out his vessel, the exploration vessel Nautilus, pictured here, along with a dual ROV system where the upper one in the yellow is called the Hercules. That's the main workhorse that gets all of our main video from the bottom. But then floating above it and also acting as the clump weight is the Argus, and it gives us kind of the top-down view from, you'll see some images later. So we went out, and what's really cool about this system, we went out in 2018, by the way, October of 2018, is that everything that we were exploring and that was being televised was being transmitted over the internet live. So everybody in the public who could find the website logged on and followed along with us. So discoveries were made simultaneously with the public. Now, of course, as a representative of the sanctuary and OET, you have to be careful with what you say because everything is being viewed by the public and recorded. So when things are exciting, you got to choose very good vocabulary and not offensive stuff. So anyway, that's kind of a fun challenge in itself. But this stuff was all transmitted via satellite back to the mainland. This is the main workhorse, the Hercules, to give you some scale. There's some people in the background. It's kind of a Swiss Army knife. You can outfit it with a variety of tools and equipment to satisfy the needs of your dive and your mission. So there's everything from manipulator arms to grab things to bio boxes to put large objects in. There's a slurp gun to slurp up gelatinous animals, and they get stored in these cylinders so they don't get shredded. There's core pushes, so you can take sediment cores. There's a whole variety of instruments to sample animals and geology and other things that are appropriate for that mission. The only downside of it is if you are targeting a certain type of animal or certain type of samples, and you come across a different type of animal or samples, it can be hard to sample itself. But at least you have the video and you can go back and try and get it. This is just a series of images showing how we launch off of the side of the ship, launching the Hercules first into the water. And this is in the control room itself, which is on the ship. So again, this is a remotely operated vehicle or vehicles. We are not inside of them. They are robots. So we don't have to worry about human lives down below. We are up sipping coffee or Diet Cokes or whatever we have. And we are watching all of the images come in live on those 4 screens in the background. In the front where it says Chief Sci, oh, you see the water bottle there, that's where I would sit. And in front of me, I would have a variety of screens of information from, depth, pressure, temperature, salinity, oxygen levels, all of that kind of stuff, kind of heading. We'd have a live map with our positioning that I could do that or manipulate with. We had a chat room because there are a lot of experts that would tune in live into a special science chat room where it was a very it wasn't a public channel, it was a private channel. So we could text back and forth with instant messages to people who were experts in something that we encountered. So it's a really, really cool system. We could virtually bring experts with us. And here's a view that I took from my actual position during an actual dive, taken right at midnight. You can see the clock there at 0000, and that's of course Greenwich time, UTC. I thought it was kind of a cool photo. And then in front of us, you can see a chat room and some of the different variables with graphs and stuff like that, along with the images from the seafloor. So it's a little mini NASA control center, to be honest. Um, when we went down there, one of the advantages of a remotely operated vehicle is that they can be down for uh, almost an indefinite period of time. When you use humans in a submersible, you have the safety considerations, but you also have just the human limits. You have a certain amount of oxygen you can bring, and people can't work for more than 8, 10, 12 hours before they get fatigued. So this is a way we could have 24-hour operations. We had 3 shifts, 3 teams of people, and we'd rotate in 4-hour shifts. So you'd be on for 4 hours, off for 8, on for 4, that kind of thing. So it worked out really, really well. Um, this is a video at the end of a 30-hour dive, which was really, really cool because we were planning to pull, and this is what happened. Now, we did test the connectivity before I came on. We did not test this video, so I'll leave it up to Susan to tell me if there's an issue with hearing or watching this video. If there is a problem, I will send the links later and we can move on. But I'm going to go ahead and play this video, and I'll just watch the chat box or listen for Susan to see if this is working.
It's working. Should there be sound? Probably not.
Okay, do you not hear sound?
No sound.
Okay, no worries. So I'll send that later. And
Oh, no,
basically,
I wouldn't necessarily say don't view it. It's pretty remarkable. So even without sound.
I'll describe some of this and I'll kind of skip forward. We found some of these octopus in these pockets of rock. Now, normally octopus are solitary. And we said, well, why are these octopus together? Well, we realized when we sat down at that first group that we zoomed in, we saw eggs under them. We also saw shimmering of water coming out of the seafloor. We said, this is odd. Let's go downslope and look. That's what the video you see right now. This is going
Wow.
downslope. And I think it does not take a scientist to understand that these octopus are aligning with geological features on the seafloor. These are not random distributions of what would normally be solitary octopus. So we realized there was something going on here. And I'll skip a little forward. You can see more and more, hundreds and hundreds of these octopus. By the way, the sound right now is us in the chat room talking live to the public and to each other. So we're like giddy young kids,
Oh,
just,
wow.
geeking
Oh,
out. So we have this group of octopus here on a flat area that we could actually land. So I'll fast forward a little bit. and we could actually land and zoom in. And I don't know if you can see it over the internet, but they're shimmering.
Oh,
The
yeah,
shimmering
yeah.
that's happening here is just like the shimmering of a hot summer day off of the pavement. And that's, of course, for the light being bent through differing temperatures of water because they're different densities. So light will penetrate it and bend in different ways. So we realized probably hot water was coming out, and these octopus were in these unusual positions, this flower-like position, and they had eggs under them. So they were all females. And so that was one reason. There's a shot from the Argus, which is the ROV above the Hercules, which gives us this unique perspective. And so this is us taking off from the site, realizing how an amazing discovery this was.
Hmm.
The problem was here, let me fast forward here. There we go. There's a good shot of that little pool. The problem was we wanted to come right back and explore this area. The ROV basically broke. The fiber optic cable inside of the bundle got severed somehow. And this is the very beginning of a 12-day mission. And so, unfortunately, we had to scrub everything and we lost the rest of the mission. And so it just left us wanting more. And that's what happened. I'll get into that in a bit. But what we realized is this was a very unique area. No one had ever discovered something like this before. And not only that, but we discovered what we assumed at the time and was confirmed later was warm water escaping from the seafloor. So that suggested, volcanic activity or something of the nature. So we were really excited. We wish we could have come back right away. We couldn't, but we did eventually, and I'll get into that. This is why we called this the octopus garden. This looks like a
Hmm.
bunch of octopus flowers in, the sediment in your garden. So it was really, really cool. There's just a kind of a close up showing you now every little nook and cranny and kind of neighborhood, if you will, of octopus are different. But this is kind of indicative of a typical neighborhood where you have a bunch of these mother octopuses upside down in this brooding position. They have eggs underneath them. They're protecting them from predators, which are shrimp and other animals that can come by and eat them. They also use their siphon and jet water that is full of oxygen over their eggs to oxygenate them. At the same time, you see this neighborhood of other animals. You have the obvious anemones, you have snails, little whelks in there, and you have some shrimp, which are hard to pick out here, and that's okay. There's a lot of video of shrimp I'm not going to show you tonight, but shrimp wait around the peripheries of these mothers, and if the mother dies or moves off, the shrimp will come in and attack the eggs. We've seen that happen. So this is a whole little ecosystem here. And if you go just a little bit off of these little neighborhoods, it's pretty much just bare rock. So this is a little ecosystem that requires a lot more study. So there's a lot of scientific study potential here in these areas. So unfortunately, we couldn't come back during that 12-day expedition, which was a real bummer. The good news is that was October of 2018. In March of 2019, the BBC was doing an event, a live broadcast called Blue Planet Live to the UK and Canada. And long story short, I was invited to come along the research vessel Atlantis, which was the home base for the Alvin, which of course was used to discover the Titanic in the 80s. That's an image of it on its 5,000th dive. I wasn't in it at that time. That was an older image. But I was invited to come along for 4 days and to dive in the Alvin, and I got a chance to go face to face with our discovery and the octopus mothers down there. So it was an incredible opportunity. We not only confirmed additional octopus populations, found thousands of them, but we actually had a thermometer we could deploy, and we confirmed that the water coming out of the seafloor was in fact warm. Now, normally water at 10,000 feet. I didn't even mention the depth. I forgot about that. This is over 2 miles deep. Okay, 3,200 meters, about 10,500 feet deep, 2 miles. The ambient temperature down there is just above freezing. It's about 34 degrees Fahrenheit, 1.6 degrees Celsius. The water coming out of the seafloor wasn't hot, it wasn't even bathtub temperature. It was in the mid 50s. That sounds cold, but it's a good 20 degrees warmer than ambient. So to these octopus, it is a bath, it is a spa, it is warm. And the thing to note about it is things in the deep sea grow and reproduce extremely slowly because cold temperatures reduce metabolism, reduce growth, reduce reproduction to a point where a lot of these animals in the deep sea live for decades, if not centuries, certainly some of the higher vertebrates like Greenland sharks. But these octopus, which I'll get to a little bit later, reproduce a lot more slowly than their counterparts in shallow water, which of course is where it's warmer. So we found some more areas we call the Octo Spa, the Octo Cone, 6 miles away. And in total, we ended up finding about 20,000 mother octopus. And on this BBC expedition, we actually witnessed the first live hatchings of some of the baby octopus. And these are just screen grabs of some of those hatchings, which was fantastic. Um, oh, this is just a funny video. It's tradition. I'll mute it there. It's tradition after your first Alvin dive to get dunked with cold seawater. So anyway, that was fun. Um, this made a lot of news in the national, headlines and international headlines. Of course, Woods Hole, Ocean Exploration Trust, Smithsonian, all these places picked it up and pushed it a lot, which was fantastic for us. And then what happened, and I forgot to mention this. The October cruise in 2018, which was scrubbed, most of it, because of technical failures and technical problems. Bob Ballard himself came aboard when we docked back in Los Angeles, came aboard, shook my hand, and said, "You get a do-over." So he was very gracious in giving us basically a free shorter cruise in 2019, exactly 1 year later in October. Now, what's cool about this cruise is we went back to the Octopus Garden, discovered even more than we had with the BBC. But when we were going over to what I named the Octocone later, they said, "Hey, Chad, do you want to stay along the seafloor, or do you want to save some time and cruise over in the midwater?" And I said, it's a bunch of mud, but let's just go over the seafloor. Well, as you can see with this teaser image here, this is what happened. And again, I know you guys can't hear. Trying to mute it here for myself. But basically, we came across a whale fall, and I'll skip forward here. We estimated this whale fall to be about 4 months old. As you can see, it still has some fat, some baleen, some cartilage. It still has some heart tissue there. These are scavengers, cusk eels and octopus and other animals still feeding off of the flesh. Now you see the rib bones there. They kind of look hairy or fuzzy. This is what you call bone-eating worms. There's a good shot from the Argus above. This was actually a adult minke whale. It's only about 15 feet long, not very long. They're very small whales. But what was really cool about it is, let me fast forward here. There, you can see the fuzz there on these bones here. These are bone-eating worms, and I have some shots here I'll show you in a little bit. And that is a very uniquely ghastly kind of discovery made by Monterey Bay Aquarium Research Institute in 2002. But it's a specific species. Actually, there's been more than 20 species named now. I should say genus of bone-eating worm called Ossidax that actually burrows into the bones of mammals, mostly, can be reptiles too, on the seafloor, and they inject these roots into the bone and suck up these lipids from the bone itself. They have a symbiotic bacteria that lives within their lining, and the symbiotic bacteria then metabolizes said lipids. Their byproduct is then absorbed by the worm itself. So the worm actually does not have a gut system. It does not have a stomach. So that's why it requires this symbiotic relationship. So the bacteria get their food, their byproduct feeds the worm, and the worm provides the shelter for the bacteria. So it's this really kind of zombie, ghastly kind of, a symbiosis between the two. But it's pretty unique to these marine mammal whale falls. Here's some shots of the whale fall itself. And what's really unique about this is that only about 75 whale falls have ever been discovered in the world. And this is probably one of the freshest ones that's ever been cataloged. Here's a close-up of those bone-eating worms coming out of the bone itself. And then we got to go back in 2020 in October, exactly to the week, 1 year later. And you can see the difference between how fresh it was, estimated 4 months old in 2019, to 2020, where everything had kind of fallen apart. So these things don't last very long, but they are a boom of calories to the deep sea and a main source of calories in the area. In fact, it's estimated to be about 200 years worth of food all at once, can last up to 100 years or more. And this is talking about just the bones. And that's where most of the whale fall discoveries have happened, just the bones. Um, and like I said, 4 months old, 75 documented in the world. And, in shipping lanes, they're going to get hit more often. I'm sure you guys have heard, and, this society, the Cetacean Society, knows about ship strikes. So, animals are hit, whales are hit, and a lot of them sink. Some of them, float and get beached, of course, but a lot of them sink. So it's quite reasonable to believe that there are more whale falls below shipping lanes. This turned into a great discovery for sanctuaries. They developed this wonderful poster series for creatures of a whale fall to look at the ecology and kind of life stages of a whale fall itself, including the animals that exploit the resources there. Working with Ocean Exploration Trust, we collected at least 2 different morphs of Ocedax. We definitely have 1 new species. We collected bones, worms, baleen, environmental DNA, and we did a lot of video orbits to produce a 3D model as well. And if you go to sketchfab.com and look up whale fall, you'll find this, where you can use your computer and spin it around, zoom in, zoom out, and see an actual 3D model of the whale fall I just showed you guys. So I won't do that now in savings of time, but this is just a screenshot from that 3D model. So that turned into a lot of extra work we did with Monterey Bay Aquarium Research Institute. Jim Barry, who's a deep sea expert there with ocean acidification, amongst other things, wanted to go out there, and he actually, kind of asked me permission. And I was like, well, I don't own this. Of course, just as long as you take me with you. So we had been working with Jim for years, working on corals at Sur Ridge and Davidson Seamount. So of course, we were able to go out with him. They have a different ROV, the Doc Ricketts, after the famous biologist here in Pacific Grove from the early part of the century. We use their vessel, the Western Flyer, which has a moon pool in the middle. It kind of opens up like that, and we would launch the ROV there. Very similar to the Hercules in terms of what it can do with its tools. And you guys are probably very familiar with MBARI. This is a shot from inside the control room where we have people up in front operating the ROV and the cameras, and then people in the back recording science or checking their phones, whatever it may be. We have a lot of work that we would do. A little bit more, I would say, the dirty work. So with the Nautilus, we don't generally take, we definitely take samples, but we don't take samples of animals per se. It's more geology and other things. With MBARI, we could actually sample the animals. Now, granted, the animals that we sampled were dying or dead. So that picture of an octopus going in the canister was a dead female. The one thing about octopus, if you don't know this already, no matter what species you are, once the female lays her eggs, she is never going to feed again. She is going to take care of those eggs, and once those eggs hatch, hopefully they hatch first, that female will, we call senescence. It will senesce and pass away. So we made it a point to not collect live females. We didn't want to do that. So this was a already deceased female, but we wanted to be able to have some of the tissue for diet analysis and other things. This is just a picture of Davidson Seamount and some of the octopus here that we found that went into an eventual publication. Here's a photo mosaic. They sent down an ROV with a camera on its belly to just mow the lawn and take pictures of all of the octopus in the area. And it's pretty easy to resolve it. They're kind of gray to white octopus off of a dark basalt background. So the computer could actually count the number of octopus, and that's where we come up with the number of 20,000 octopus in this area, which is pretty remarkable. We also had the ability to leave long-standing recording equipment. So we could record temperature, we could record oxygen, we could record a few other things down there. What we noticed is that temperature and oxygen fluctuated along with tides, so it's tidally locked, which is pretty cool. That something that deep is actually still influenced by the tides of several feet up above, even though this is more than 10,000 feet deep. Um, this is getting to the point of why this is so important. So, why does this all matter? Well, I kind of alluded to this before. Things in the deep sea take a long time to breed, to grow, to reproduce. Things in warmer waters, which is typically your shallow water, things happen much more quickly. The giant Pacific octopus, which is one of the largest octopus in the world, we have here in Monterey, they only take 3 months to brood their eggs, and they're much, much bigger than these octopus in the deep sea. Well, a similar species to this, which is the pearl octopus, this is called Musoctopus robustus, another similar species that Mbari caught while she was laying her eggs, were able to go back and visit her over years. It took her 54 months, 4.5 years, until her eggs hatched. And that wasn't even at as cold of a temperature as these octopus were at. So if you look at this growth curve here, this is a plot of all the octopus brooding time or brooding periods with their ambient temperature. That red dot there basically represents what these octopus would be reproducing at this temperature, or the one that they followed. The blue dot is the theoretical time it would take for one of our octopus to hatch her eggs, over 10 years. 10 years, which is insane. We found through our work with Mbari, it took under 2 years. And so I'll get to that in a second. Here's some more work we did with some stop motion cameras that we left down there for a period of months. And here is a result of that time lapse photography. And it doesn't look like much at first, but if you pay attention, you can actually see obviously brooding females here. And you could see the anemones waiting for a meal, and you see other females in the top right. Eventually, one passes away and moves off, and then another female comes in to take her place. There goes the female. Now the eggs are being consumed by the snails. Now it's blank, and now a female comes in to lay her new eggs. So this is kind of the life cycle of each of these areas. These females somehow know to come back to these warm water areas. And so essentially what we've determined through our work with Mbari and what we published in Science in 2023 was that this is a cheat code. This is a cheat code for the octopus. And they're going from basically anywhere between 6 and 10 years down to under 2. So this is a reproductive strategy that they figured out through their evolution. If they find these warm water areas, it will speed up the metabolism development of their eggs, but it also reduce the predation time that those eggs could be preyed upon. So it's kind of a win-win for them. What we don't know today is, we see these little babies hatch and fly off, swim off into the distance. How do they come back here? How do they find this area? Do those same babies come back? Is it random? Is it just moths to a flame kind of situation? Is it a chemical signature? Is it magnetic? We don't know. We just don't know. There's so many questions. Every discovery generates so many more questions. And so there's a lot more work to do here, but it's been a remarkable discovery. Now, working with Mbari over the years, we worked with the BBC again. And in the picture that I showed you, there was a producer from the BBC, Will, who came along through 7 or 8 of our cruises, and they were looking for content for Planet Earth 3. Now, I mentioned to you earlier, we had work at Davidson Seamount in 2006 that published or aired in Planet Earth 1. Well, this octopus garden got a 6-minute segment in Planet Earth 3 in their ocean segment in November of 2023. The video I have here is that 6-minute segment, but because I don't have audio for you guys and I'm running a little bit over, I'm not going to play it. So I will try and send that link to Susan, but I do have to be careful about the copyright issues there. But for those of you who do have the BBC or do have Planet Earth 3 and have not seen this, please look it up, because I will tell you something. Hearing David Attenborough narrate your discovery on the BBC is pretty freaking cool. So that was an amazing little nugget that we were able to kind of extract from that. And again, I'm not going to show you this because of the audio.
Oh, wow.
Remarkable cameras. that they were able to use with Mbari, with lights, dramatic come overs, these ridges. There we go. You get some of the really great BBC style close-ups of the eggs, and David Attenborough's narrating everything right now. Wish you could hear it. So it's a really remarkable segment from Planet Earth 3 that I really, really encourage all of you to check out if you can. So I'll move forward. Um, So I want to transition into a little bit of work because I have a little bit of time left, about 10 minutes or so, because this is the American Cetacean Society, and I haven't talked too much about cetaceans, I've realized, but I wanted to bring you a lot of that deep sea work. As I wrap up some of the deep sea stuff, this is just some of the imagery that really, generated the grassroots level effort to get Davidson Seamount included into Monterey Bay National Marine Sanctuary. I was telling you about the sofa-sized couches or sofa-sized corals and sponges. This is kind of what we saw down there. These things are very, very large. I got to do two dives in the Alvin. One of them to the Octopus Garden, but the other one was to the summit of Davidson Seamount. So we got to park the Alvin underneath some of these corals. This particular coral is called a bubblegum coral. You could probably understand why. It's pink. It looks like bubblegum. And these things really were just as big as the Alvin. 10, well, Alvin's bigger than 10 feet, but looking at my porthole to see something that was much wider than I ever envisioned looking through a TV screen. Pretty, pretty cool. All right, let me go ahead. All right, so I'm going to wrap up in the last 10 minutes here some of the work we've done with marine mammals. So I do lead some of our offshore research here, and we've been doing this since 2010. 2010, we used the MacArthur, which is a NOAA fisheries survey vessel. But in 2015, 18, 22, and 24, we used the Bell M. Shimada, and we have another cruise scheduled for this July in 2025 aboard the Reuben Lasker. This is a wonderful platform that we can use to do a variety of work, but our main objectives for our work over the last decade has been to conduct visual surveys along these transects over the seamount to collect abundance and distribution data for seabirds, marine mammals, and other wildlife. We also drop a CTD, which is conductivity, temperature, and depth, so we can get kind of the profile of the water column down to 500 meters. We also collect water for environmental DNA, and more recently, we've been doing microplastics. I'm leading a separate project, which I won't get into, called Watershed to Whales, which is looking at microplastics in rivers, beach sand, ocean water near those rivers, and then even ocean animal stuff, including fecal material from whales, fishes, and krill. Also, when we're underway, we collect acoustic data, so kind of the prey that these marine mammals will feed off of with our echo sounder, and we ground truth it with tows, with a tucker toe and a hoop net as well. Although we're not able to do that in 24, and we won't be able to do it this year either. And we have also done the microplastics work as well. So here's a map showing how we cross over the seamount perpendicularly, but conveniently. It is also designed to be against the northwestern predominant swells, so we're not in the trough and get as seasick as we probably normally would if we went the other direction. And there's a picture of some of our crew from just this past year. By the way, I saw a couple names in there from our crew. So hello, everyone. I think I saw Lori in there. And anyway, that's our marine mammal observers, because I'm not an expert at this. I'd go, oh, that's a whale. That's a bird. These people know exactly what species they are, what plumage they are, where they're from, all those things. So we hire the professionals to help us out. So they were up on top to do that observations. And there's a picture of the ship itself in San Francisco. This is the CTD, which with a rosette of Niskin bottles, which we would sample ocean water from different depths. You can basically trap the water at whatever depth you want. So you can drop it down to 500 meters or even deeper, and then water from 500 meters will be in those columns, and then you can close the top and bottom and trap that water. So you can sample the chemistry from it, but also the environmental DNA from it as well, which we worked with Mbari on doing. We have a live kind of picture of oxygen, salinity, temperature, and depth up in our control room so we can kind of see the progress of that. Just a picture of some of our scientists talking about before a net tow we were going to do. This is in 2022. So we have the ability to kind of plan. This is everybody geared up, ready to launch those nets. And again, this is more of the ground truthing of the echo sounder. We want to sample the prey. Now, at nighttime, there's something called diel vertical migration, where a lot of these animals that are hiding from the sunlight, they don't want to be visible to predators, will come up in the nighttime to feed. So, well, it's easier to sample them when they're closer to the surface. So that's when we do sample them, is in the middle of the night. So we'd sample them with these massive nets, drop down to about 300 meters or so. We come up with a whole variety of animals, from the most common pyrosomes to fishes and all sorts of cool stuff. And we would kind of separate them into their own jars and classify them. And you can see the amount of jars that we could kind of accumulate over a period of time. These are the marine mammal observers up top in the observation deck. And this is the kind of results we would get, just kind of graphically. Here's some bird and marine mammal data on the two areas. These are the transects that go across the seamount. Way too much data to get into for this presentation, but just giving you a flavor of it. And then they've sent us out a couple times in May, which isn't very fun. This past summer, or this past May, we actually saw some Biggs killer whales. And you guys probably know a lot more about Biggs versus resident killer whales than I do, but this is a very big deal back in May. And so we were able to capture some video and photos of him, and they were able to send this information out. You guys probably know the differences much more than I do, but of course they're separating Orchinus orca into two different species. So do you still call them orcas? I don't even know anymore. Anyway, so it's really interesting to kind of come across those big killer whales. Of course, we come across fin whales, which is probably one of the more common whales we come across too, along with blues. Not just dolphins and other marine mammals and sea lions, but also mola molas and things out there. And then, of course, all sorts of birds, from shearwaters and albatross and fulmars and more exotic birds as well. Just a few of the shots there. It can get quite nasty out there in May, so it does become problematic to a lot of the work that we want to do. This is a good kind of clip of some of the conditions we face. There's lots of that. And so anyway, just some more pictures here as I wrap up here, as we're getting to 8 o'clock. We also did environmental DNA the last couple of cruises. So Erica, who is in the lower right here, my colleague here at the sanctuary, and I will handle all of the environmental DNA processing with a little mini lab we have. So we sample the water and stabilize it, and then Mbari will analyze it. And then just some of the imagery coming back in and out of the Bay Area. We usually come out of San Francisco, so it's a very cool tourist vibe to go under the bay and Golden Gate bridges when the weather is nice. You can get fog bows there and all sorts of animals to greet you, and it's just a wonderful crew to work with. A lot of fun. Play board games at night and just have a good time as you're doing science. 22 hours of the day. So. Anyway, I know I was supposed to wrap around 8 o'clock, so I think I hit that mark. But if anyone who is not asking questions tonight can certainly contact me at that email there, chad.king@noaa.gov. And then Susan had mentioned the Simon program. So we do have a website, sanctuarysimon.org, where we also have a photo database, and we have an app on iOS called SeaPhoto, where all those photos that we've been taking over the years are available as well. There's so much more I could show you and tell you about, but I'd be here for 3 hours. So anyway, that's going to do it for my presentation, and so I'll just turn it over for questions.
Awesome. Thanks, Chad. That was incredible.
For the additional time.
Yeah, so I mean, that was fantastic. We have a bunch of questions coming in. I'll try to categorize them and synthesize them so we're not too far all over the place. First one, that's a few people ask stuff like this, and feel free to decline to answer any part of it if you're not comfortable. But so about what percentage of NOAA or general deep sea expeditions are publicly funded versus privately funded? And on the public side, which I assume is the majority, is there any fear of disruption, or how are you thinking about funding given the new administration and everything?
Mm. Yeah, you know, I could not answer the. Yeah, you're right. It's going to be tough to answer these. First of all, because I just don't know the percentages between publicly and privately funded. Because, for example, when we were use the fisheries survey vessels, so every year I apply to that, and that's an internal thing. NOAA has a whole fleet of these fishery vessels. They have a number of ships that they run. They're obligated to run. They have a regular budget. We apply for ship time. So whatever ship time we get is not additional money that's being spent by them. It's money that was already being spent. We just got selected to get that ship time. So that's a bonus to us as an applicant, and it's not. It is government paid, taxpayer paid, so it's not outside the government. Things like Ocean Exploration Trust, that's a private organization, and so that's an agreement between sanctuaries and OET, where Sanctuaries is paying them some certain money, OET is subsidizing some of that cruise. So that's a kind of a different arrangement. I don't know the percentages overall. That's above my pay grade. Yeah, unfortunately, the new administration, I'm sure everyone has seen, there's a lot of things going out, and so it's been a whirlwind and a fire hose of stuff, and all I can say is I can't comment on a lot of it, but it's going to be probably a period of adjustment as we understand what the ramifications are going to be overall, to NOAA specifically and to science. We really just don't know.
Got it. Makes a lot of sense. All right, now onto more fun stuff. So on there's a bunch that are kind of generally in the octopus garden realm. First one like super high level. So obviously a lot of this was kind of octopus focused. Are cephalopods, or I guess like mollusks more generally, because you were talking about snails too, specifically well designed to live at these depths, or are you just kind of more focused on that area of study?
Yeah, I mean, invertebrates as a whole are going to be more adaptable to the deep sea. Fish tend to have specific proteins and enzymes that will fold under certain depths that invertebrates won't. That said, we're not at crazy. I mean, we are very deep depths, but not Mariana Trench. Like, you know, the deepest part of the ocean is like 11,000 meters. We're at 3,200 meters, so we're basically not even a third of the way to the deepest part of the oceans. That said, yeah, fish get less abundant as you go deeper. These animals, like I was saying in the beginning, we were just looking for corals and sponges and those kind of invertebrates, and discovering these more advanced, you know, intelligently, I guess you could say evolutionarily, cephalopods and octopus, was quite remarkable, because even though they aren't schooling in terms of communication or working together, it was more like moths to a flame. Um, it was something that just no one ever expected. And I think the lesson that I've gotten from this is I expected this to be a fairly, you know, I know it sounds weird, but a boring, quote unquote, expedition. Not that deep sea exploration is boring, but when you see, you know, mud flats and very little corals and you're talking amongst your colleagues, it's not exactly a rock concert. So there's little excitement here and there for animals, and you know, we'll come across, you know, a Dumbo squid, or you know, a little shark that swims by, and those are our great little, you know, blips. But something like this is just highly unusual, and we've been very fortunate not only with this discovery. What I didn't mention was when we had that breakdown of the ROV, we went down to Scripps to repair it. Unfortunately, they had to cut the cable in half, which went from 4,000 meters to 2,000 meters, meaning we could not go back to the depths we were at Davidson. However, we tried to go up, got blown out, came back down to the Channel Islands. We dove in an area that we ended up discovering a large glass sponge reef. And that actually became a huge discovery in its own, and it turned into a whole different project. So what all of my deep sea research and exploration has taught me is, first of all, you don't have to be an expert in this stuff, and you can discover stuff just by pointing on a map and going, let's go there. But you never know what you're going to find when you go out there. And I think that too many people have an assumption that we've discovered or we've explored so much of the deep sea. And this is just proof that 2 out of 3 dives that I've done in the deep sea has revealed something remarkable or never discovered before. We have so much left to explore and so much left to discover, and it just begs on everyone, we got to protect these areas. We cannot exploit the oceans. We have to understand that so much is out there that we could be inadvertently destroying through our actions, and we just need to know what's out there.
Awesome, thank you. Um, and then as far as scale, can you talk a little bit about the size of the octopus you were seeing? Like, but it's a little bit easier to tell at
Mhm.
the whale fall, but in like the octopus gardens, can you talk about, how big those anemones were and how big the octopus were?
Yeah, the octopus are about softball size, about 10 to 12 inches in diameter or so. So, you know, about yay big or so. I should probably stop sharing on my screen. Um, kind of a little bit bigger than your fist. The when they spread their arms out, they're quite a bit larger, but they're small compared to the giant Pacific octopus. So it's pretty remarkable. The other thing that we notice is they have chromatophores. If you notice, they didn't change color like a lot of octopus do. And the conclusion, or at least the assumption, is in the deep sea, there's no light, so they don't really need the chromatophores to change color, but they still have them from evolution. They're just not very usable, I guess. They're not functional, if you will. Kind of like a lot of animals who have eyes in the deep sea that, might resolve a little bit of light, but they're not really needed.
Makes sense. Um, and then I think you mentioned when you kind of found the warm water with that, formed those gardens. Did the analysis of the minerals or anything kind of give you a hint to what's the source of the warmth?
Great question. Yeah, I didn't get into that. So we thought maybe there was some latent volcanism happening. Not that we thought the seafloor was going to explode anytime soon, no Vesuvius or anything like that. But when we started working with experts, we determined that it was what is classified as a low temperature ridge flank hydrothermal system. That's a mouthful. Uh, Basically, if I were to bring up a map here, I won't right now. If you think about it, the basalt rock, the volcanic rock that we see down there at Davidson Seamount, because it is a volcano, is porous. Like, when you go to Hawaii or wherever that has a volcano, you pick up a rock. It's a little bit lighter, although this basalt's a little heavier, but it's got pores in it. It's got air chambers. It's got channels in it. So that is the conduit in which water can flow through. Well, sediment, the thick mud that deposits, over eons over this rock, water cannot flow through very easily. So Davidson Seamount itself is this big volcano where water can kind of percolate through and down. It'll go through these subterranean chambers and channels of volcanic rock underneath sediment. Well, as it travels underneath those subterranean channels, just because of sheer pressure of hundreds of meters of sediment above it, as you increase pressure, you will increase temperature. That's just physics. So that water will actually be warmer than the ambient seawater above it. It will find its lowest pressure valve, just like, a tube of toothpaste, and it'll come out these little channels of rock, which is what we found off of Davidson Seamount. So Davidson Seamount itself is a big thing of rock, then a bunch of sediment around it, and these little outcrops, these little islands of rock that peak up above the sediment. So while that water is traveling through the seamount, underneath the sediment, and then out these little channels of rock, which is what we discovered. That water's been slightly warmed, 20 degrees or so warmer than ambient. The chemical signature, however, is not that much different than the water that came through. There's a little bit of a change in magnesium and calcium, and a few other elements, but nothing dramatically. So it's really, we think it's just about temperature, that the temperature accelerates the development of the eggs. And that's what the octopus have learned over probably eons, through evolution and adaptation and instinct, is that if they lay their eggs in warm water, their eggs will hatch sooner, and that will increase the throughput and reproductive success of their species.
Awesome, thank you. Um, And then as far as predation, are there any predators for the octopus in those in the gardens, or I guess in the whale falls, or anywhere at those depths?
We don't know about the octopus themselves. We've never witnessed any predation. We do think that the adult octopus are probably pretty safe from predation. Not that, you know, some sort of deep species of shark, although there's really no sharks in that area that would take advantage of it, like a Greenland shark or anything like that. So for the most part, the adults are predator proof. Their babies are, their eggs are, and so that's the whole reason that they cover them. That's the whole reason that they shield them. So uh, I didn't. There's some videos we have. I can send these links to Susan, who can send them out later. There's a whole list of videos I can send you guys, but one of them in particular are these shrimp that I mentioned earlier that hang out on the periphery of these eggs. And God knows they probably sit there for days, weeks, months before they get an opportunity. But once a female either dies or moves off her eggs, those shrimp will come right in and eat those eggs. So that's the secondary thing. If you can imagine a female who might take 6 to 10 years for her eggs to hatch versus just under 2, that's a lot less in terms of predation pressure as well. So yeah, we don't think the adults are preyed upon, but their eggs are definitely preyed upon, and so that's why they protect them.
Makes sense. Thank you. Um, all right, and then I guess a few more on the non-octopus bit. Well, one, we have obviously a very whale-focused group. Have you happened to share your killer whale photos with the California Killer Whale Project in Monterey?
The people that were aboard the cruise, they were all the marine mammal aficionados. They shared it far and wide. I mean, with many, many, many different channels. I am sure they shared it with them. I couldn't say for sure. I don't have the email with me. They were beyond excited. They had news reports on it. They had thousands of people that responded. So there's no doubt in my mind they alerted all of the appropriate people because it was a huge public, uh, you know, uh, output, I guess you could say, from the cruise for sure.
Love it. Sounds like they were all over it. And we
Yeah,
just had one come in back on octopus. What's their diet like when they're brooding?
they don't eat. They don't eat at all. So once they lay their eggs, they're done. And so that's what's really remarkable. I mean, this is the definition of a dedicated mother. They once they lay their eggs, they've already had their last meal. And these octopus mothers sit there for almost 2 years not eating. And that's part of the senescence um, kind of biological cycle. They expend the rest of their energy on taking care of their eggs. And that's why sometimes we do see the female mothers die before their eggs hatch. It's rare, but it does happen. Generally speaking, the eggs will hatch, then the mothers will die. It's called senescence. But they don't eat anymore. I mean, they are fully dedicated to making sure their eggs are protected and hatch.
Makes sense. And then can you talk about? Sorry, a bunch of new octopus questions came in. I think people were upset that I said the octopus part was over. Can you can you talk about how any variations among different species within the octopus gardens, or are they all kind of the same species? And if they do you think they're genetically related from like a familial point of view?
They're all the same species, Musoctopus robustus, and that's called the pearl octopus. It's actually a cosmopolitan species, meaning you'll find them in every ocean of the world. So they are not unique to our area. They are found off Japan, off Africa, in all the deep sea areas of the world. It's just that they're solitary. They're normally found brooding on a random rock in the deep sea, near no one else. This is only the second time they've been found grouped with other octopus. The only other situation they've been found was in Costa Rica prior to our discovery. However, at the time there was only about 150 individuals, and we've cataloged 20,000 here. Since our discovery, they finally have documented a couple of live births or hatchings of eggs down in Costa Rica, but the number is still far fewer than we have near Davidson. What I'm excited about is the fact that we have found these species near rocks that are basically conduits for deep sea, rivers. water coming out that's slightly warmed. Most people explore in the deep sea. So you have to remember, everyone, that deep sea exploration is rare. It's very expensive. It's very hard to do. And when you do get out there, it's often canceled due to weather or technical problems. And so what we find ourselves doing is exploring the areas where we know we're going to find beautiful, amazing things. Like I showed you at the top of the Davidson Seamount, corals and sponges, right? You'll find most of the cool stuff at the tops of rocks, at the tops of ridges, at the tops of mountains, those kinds of things, because that's where all the currents come up. That's where all the corals bloom. You won't find them down in the deep where sedimentation is high, where things are buried, where flows are low, where currents are low. We've now discovered that this is not necessarily the case. So my theory is that, and according to the hydrologists who are deep sea water experts, these deep sea rivers are everywhere. There's all over the world's oceans, water is coming through basaltic rock and exuding out somewhere else. But it's all in these flat, relatively innocuous areas that don't look very exciting. So my theory is if we start exploring these areas in the, kind of foothills of seamounts and the foothills of mountains, if you will, we're going to find that this is a very common occurrence, that this species, I mean, think about it. This species that has existed for millions of years, there's no way that this is the only place on the earth that they do this. If this is an evolutionary trait, if this is an adaptation that they've evolved to take advantage of, this is not just happening in our sanctuary. This has got to be happening, in a regional or even global context. So to me, it would be fascinating and important and imperative that we start exploring these other areas to see how this species and maybe even other animals are exploiting these low temperature ridge flank hydrothermal systems across the globe. And maybe that will spur some conservation movement to prohibit a lot of human activity in these areas, whether it be deep sea mining or what have you, which is becoming a very major thing in terms of manganese and other things like that. So anyway, don't mean to get on my soapbox here. I'm just talking as a scientist. I find it very fascinating. Something I didn't even know about more than 5 years ago is now something that probably is way more common than we actually think. It's just we're not looking there.
Awesome. No, I love the soapbox. That's why you're here. Um, And yes, that sounds good. I think I know I've been peppering you with questions for the past 20 minutes, so we'll try to keep it a little bit brief from here. But maybe we can pivot to some environmental concerns. Can you talk about if, in your experience or just kind of in the readings that you've done, what the effects of, climate change or ocean pollution have been on various kind of places and various depths, I guess?
Yeah, so I've been directly involved with a couple of things, and particularly with persistent organic compounds or pollutants called POPs. That's things like DDT, fire retardants, things that are used kind of in farmlands, inland, or whatever. They get eroded and into the rivers and streams, and then get, put out into our sanctuary in the oceans. I've also been working on microplastics. Well, in my direct work for this particular cruise in 2018 and 2019, we actually sampled the water column and even the sediments at 10,000 feet deep, and we detected those fire retardants and DDT at those depths. Microplastics we discovered at every surface sample we've ever taken. MBARI has discovered microplastics in every sample they've taken down to 1,000 meters. And now I'm a part of a state-funded program called Watershed to Whales. I mentioned that earlier in the talk at the beginning, where I'm looking at sampling microplastics from rivers, beach sand at 4 rivers around Monterey Bay. That's San Lorenzo, Pajaro, Salinas, and Carmel rivers, and then ocean water offshore of those rivers. And we've discovered microplastics in every single sample of every one of those locations. And so the conclusion that I have, just scientifically, is microplastics, DDT, fire retardants, other persistent organic compounds, they're everywhere. They're ubiquitous in our environment. The question is, how is it affecting our health? I'm not an expert. I can't tell you that. Um, but that is a major concern of mine. You know, I love studying the marine environment for the animals and what they do and how they survive and what their adaptations are and their evolution. But you know, as human evolution, you know, or human civilization advances here, we tend to put a little bit more in terms of pollutants, and maybe we don't know exactly what we're doing. We all, you know, love the convenience of our plastics and our washing machines and everything else and our clothing, but it's certainly having an effect on the environment. As far as it affects our human health, I just don't know yet. I'm not an expert on that. Um, I just know that what we do has left an impact or a fingerprint everywhere we look.
Awesome. Thank you. All right, cool. I think from there, I guess we'll have to have one more on a more uplifting note because I hate to end on a pollutant downer note.
I didn't mean it downer. You know, I'm trying to. Yeah, I don't want to say like you are gonna
No. It's the I
suffer
mean that's the
because of
nature
this.
that's the nature
Yeah.
of this world. But no, is there anything that you're most excited about in the next 5 years in your research, or anything that you're the most excited to look at, or expectations for the future?
Yeah, we're going to be working with Monterey Bay Aquarium Research Institute. They're getting a new vessel. Um, the David Packard, of course, named after Packard, who started, you know, Hewlett Packard, and he was the main funding of Monterey Bay Aquarium Research Institute and the Monterey Bay Aquarium. They're getting their new vessel this year, and so we're going to be, you know, going out with MBARI again to look at the octopus garden, but also Sir Ridge. We're going to be doing a lot of good work, not only doing more work at the octopus garden, but we have been, and I didn't mention this earlier, Deepwater Horizon happened in 2010 in the Gulf of Mexico. And unfortunately, you know, the oil that was covering the surface of the ocean was, they used a surfactant, basically like kitty litter, if you will, which kind of took that oil and it sank from the surface of the ocean. Everyone said, hey, problem solved. It's all gone. Well, it sank to the bottom of the ocean and it blanketed and covered deep sea corals that died. And that was all well documented. So we wanted to look at a way, if in the future, with ocean acidification and rising global sea temperatures, which will affect deep sea corals because they live and have adapted to a very narrow range of temperature and oxygen levels. Animals at the surface have a much wider tolerance because of the variability in temperature and oxygen at the surface versus the relative homeostasis of the deep sea. And we said, hey, can we actually transplant corals, deep sea corals, from one area to another? And over the last 10 years, we have proved that. Scientifically, we've published papers on it. It's been wonderful. So we got funded to continue this work. So we're going to be working with MBARI to do some more coral transplantation with them. So I'm really excited about that, and then working with them in terms of the octopus garden, because all of the little bits of you know, the community level stuff that I was talking about earlier with the snails, the whelks, the anemones, the shrimp, and other things like that. And where do the babies go? How do they come back? How do they find their spot? Why do they lay their eggs in warm water? We have so many questions that we want to answer, which will apply to many other systems around the world. So we have a lot of work coming up. We have a lot of work down Big Sur. We have a lot of work dealing with kelp loss around not only the Monterey Peninsula, but all of the California coast. We'll be doing a lot of drone work and scuba diving work. The list is endless, but it's all very exciting. And, you know, we really want to get to the bottom of a lot of this stuff. We certainly want to answer the questions from the public and the state and, you know, the U.S. public, really. I mean, we're working for you guys. We are servants to you, the American public, and we're trying to do what we can to protect and understand our natural resources.
Amazing. Well, thank you. I'll pass it back over to Susan if she has anything closing, but really appreciate everything, Chad.
Yeah, of course.
Yeah, Chad, I think this is, we should look at this as chapter 1 of your talks to our chapter, because just like all this great research creating questions, I think our audience definitely wants to go deeper into a lot of things you touched on. But I wanted to mention, do you happen to know a Maya King?
Yeah, that would be my daughter.
Okay, well, she's got some good questions, and I suspect she has an inside channel too. But yeah,
I can answer those on the side. It's okay.
yeah. So anyway, fantastic. But yes, it's been wonderful. I've already got an idea of having you come back, so I will be in touch. But between now and then, gosh, just keep up all this great work, and the discoveries that are so great, and yet some of the things that aren't so great, for instance, around microplastics, that we really need to know about regardless, so that we, as our own species, can become a lot more conscious. So
For
anyway.
sure.
All right, well, onward in 2025. And thank you, chapter attendees, and we'll see you in February.
All right, thanks everyone. Appreciate it.
Okay, good night.